Thrombocytes or platelets are not actually cells, but rather are cell fragments. Their lifespan is about 10 days. The purpose of these little cell fragments is to release chemicals that act in the clotting process. Normally, there should be 150,000 to 400,000 platelets per microliter in the blood. Platelet formation starts with that same hematopoietic cell, the hemocytoblast, and this time the daughter cell commits to becoming a megakaryoblast. As this cell goes through its cell division and maturation, instead of developing into a cell, it splits off tiny little fragments of cytoplasm that contains enzymes, and these are what are released to the circulating blood. Platelet formation is hormonally controlled by a hormone called thrombopoietin. Platelets are involved in hemostasis, and hemostasis is the stoppage of bleeding. There are 3 steps to hemostasis. We start with vascular spasm. Anytime smooth muscle is cut, it tends to contract or spasm. So this is an immediate response to blood vessel injury. The more muscle there is in the blood vessel, the greater the spasm. The arteries have a lot of muscle, so they will spasm and almost close completely up for a period of time. Capillaries have no muscle in them, so they never spasm. The damaged tissue leads to platelet plug formation. The platelets are called to the area and begin to aggregate. Because this is only a temporary solution to the problem, we have to reinforce this platelet plug with fibrin. The 3rd step of hemostasis is the step of coagulation, or actual blood clotting. This is where we're going to produce the fibrin that will stabilize that clot. Coagulation itself is a 3-stage process that results in fibrin production. Whenever the blood vessel is cut, we're going to have the vascular spasm. That is, the blood vessel will close up in an attempt to try to close that damaged area. The damaged area will have exposed collagen, and that's one of the things that will make platelets adhere to the area. So the platelets begin to stick to the exposed collagen, and they begin giving off chemical signals that call more platelets to the area to reinforce the plug. However, we've got to have coagulation to make the fibrin mesh that will make an actual clot. Coagulation is a 3-phase process. In phase 1, we activate one of 2 pathways. The intrinsic pathway is activated by negatively charged membranes, like those found on collagen or platelets. Glass also is negatively charged, so glass will activate this pathway. The extrinsic pathway is activated by tissue thromboplastin. The intrinsic pathway is activated by things that are actually going on at the site of injury. The extrinsic pathway is activated by things that are going on near the site of injury. Both pathways lead to a common pathway, and at the end of phase 1, no matter how we got there, we have something called prothrombin activator. In phase 2, prothrombin activator converts prothrombin to thrombin. And in phase 3, thrombin converts fibrinogen to fibrin. Fibrin is what forms the stable clot. It makes a web of fibers that hold blood cells in place. Also, after a few hours, fibrin begins to retract or shorten, and this helps with wound closure. As we look at the cascade, in the intrinsic pathway, the damaged blood vessel starts this activation of factor 12, which activates factor 11, and so on down the line. On the extrinsic side, tissue factor activates factor 7, and we activate so on down the line. This is called a cascade because we have to activate one thing to activate the next thing in line. Both come together at this common pathway, which ends in prothrombin activator. So phase 1, no matter how we get there, ends in prothrombin activator. Phase 2, prothrombin is converted to thrombin, and in phase 3, thrombin causes fibrin to be formed. This gives us that cross-linked fibrin mesh that is going to hold red blood cells. And here we see that fibrin mesh with blood cells trapped in it. This is your stable clot. Now, clots can't last forever. We have to make them go away sometime, otherwise we'd be one big mass of clots. Fibrinolysis is the process that removes those clots when they're no longer needed. This also helps prevent a forming clot from blocking a blood vessel. One of the substances that's released at the time of all of the clotting factors is plasminogen, and plasminogen is incorporated into the clot as it forms. Anytime there is a clot in or around a blood vessel, the endothelial cells begin to secrete tissue plasminogen activator, or TPA. Tissue plasminogen activator will convert plasminogen to plasmin. Because plasmin breaks down clots, TPA is sometimes called clot buster. Because that plasminogen was incorporated into the clot as it formed, as levels of TPA get high enough, plasmin begins to dissolve the clot. Fibrinolysis begins within 2 days of clot formation and continues until the clot is dissolved. Once clotting starts, we want to make sure that we limit clot growth or formation. There are 2 ways we do this. One is that fast-flowing blood tends to remove clotting factors from the area pretty quickly. The other one is that we have a number of ways that we can inhibit the activating clotting factors. Thrombin, which is what activates the production of fibrin, will begin to bind to fibrin. If not, we will have a positive feedback mechanism going. Thrombin actually feeds back to the other clotting factors to keep them activated, so we make more thrombin, so we make more fibrin. So if we begin taking thrombin out of the picture as we make fibrin, we break that positive feedback loop. Antithrombin 3 in the plasma also inactivates some of the unbound thrombin. The liver produces a protein called protein C, and this can feed out and inhibit some of the intrinsic pathway factors. Heparin is naturally produced by basophils, mast cells, and the endothelial cells of the blood vessel lining. Heparin also inhibits the production of thrombin. When we have a damaged blood vessel, we need a clot, but we want to make sure we don't get clotting when we don't want it. The smooth endothelial lining of the blood vessels is one of the things that prevents unwanted clotting. As long as the blood vessel lining remains smooth, the platelets will not try to aggregate anywhere. There are also antithrombic substances that are secreted by the endothelial cells, things like nitric oxide and prostacyclin. These sort of make the endothelium slick so that platelets don't adhere easily. Some people take vitamin E. Vitamin E quinone is formed in the body when vitamin E reacts with oxygen. This also prevents blood clotting. There are anticoagulant drugs. Aspirin helps prevent platelet aggregation. Basically, the platelets don't want to stick to each other. Heparin, as we've already mentioned, blocks thrombin. Heparin is available pharmacologically as well as what's naturally produced by the body. And then there is warfarin, better known as Coumadin. This interferes with the production of some of the coagulation factors that are vitamin K dependent. Thromboembolic disorders are conditions where you have a thrombus, a clot in an unbroken vessel, and or an embolus. Now, an embolus is a clot or other substance that free floats in the blood. A clot that breaks free from the blood vessel where it formed would be a thromboembolus, but you can have other kinds of emboli, like air emboli or amniotic fluid emboli. The problem with emboli is they can block blood vessels. They may travel in larger blood vessels, but as they get into smaller and smaller blood vessels, they can block blood vessels, and that can prevent blood from getting to tissues that are further along in the system. Thrombotic emboli can be treated by dissolving the clot. We can either use TPA or streptokinase. Streptokinase is a substance that is produced by bacteria. The most common cause for a thrombosis or a thromboembolus is a roughened blood vessel. Plaque collecting on the inside of blood vessels is one of the things that can make a blood vessel rough and can encourage the production of thrombi. It can be prevented by taking a drug. A lot of people take aspirin. This keeps the platelets from congregating at some of those little minor rough spots in the blood vessel. Heparin is usually given when you're in the hospital. Heparin is frequently given during surgery when you don't want the blood clotting even though it's exposed to air. The advantage of giving heparin is that it's easily reversed. They can use a substance called reptilase, which they get from snake venom. Warfarin can be given to people who need to combat blood clotting long term. This will interfere with the formation of some clotting factors. It won't prevent them from being formed, but it will cause them to be formed in smaller amounts so that blood clotting is a little bit delayed. Thrombocytopenia is a condition where there are too few platelets, less than 50,000 platelets per microliter. What happens is there aren't enough platelets to plug even tiny little bleeds that might occur in the body, and so there is spontaneous bleeding. Depending on where this occurs, if it occurs in the brain, for example, you can have a stroke. Typically, thrombocytopenia is caused by bone marrow depression. Viral infections can be one cause of bone marrow depression. Certain drugs, particularly chemotherapeutic drugs, can be another cause. Because the liver makes a great many clotting factors, any kind of liver dysfunction may lead to a bleeding disorder. Some of the blood clotting factors are dependent on vitamin K. Vitamin K is something we can take in our diet, but we have bacteria in our intestine that make all of the vitamin K we need. So a lack of vitamin K is typically not a problem. But if there are serious liver disorders like hepatitis, hepatic cancer, or cirrhosis of the liver, there may not be enough clotting factors formed, and that can lead to bleeding disorders. Hemophilias are a collection of hereditary bleeding disorders. Hemophilia A is the most common. This particular disorder is sex-linked. Women carry it, but only male children display it. This is factor VIII deficiency. This can be treated with factor VIII. Hemophilia B is also a sex-linked characteristic. Here the factor that's deficient is factor IX. And hemophilia C is seen in both men and women. It's a fairly mild form of hemophilia, and this is a factor XI deficiency. Disseminated intravascular coagulation, or DIC, occurs when there is widespread clotting throughout the body that results in severe bleeding. There are not functional clots produced, but the clotting factors are consumed throughout the body, so that there is no way to stop bleeding. This can be a complication of pregnancy, a result of septicemia, or a complication of a transfusion reaction. Because once we start producing thrombin, we've got that positive feedback mechanism that continues to activate the clotting factors. We need to stop that positive feedback. So the treatment for DIC is to give heparin. Heparin is an anticoagulant, so it seems kind of odd to give an anticoagulant to stop bleeding, but this actually stops the positive feedback mechanism, stopping the clotting cascade so that we can actually build up clotting factors and functional clots can form where they are needed. From time to time, it is necessary to replace the blood or the fluids in the circulatory system. Whole blood is virtually never used. It's usually broken down into its component. It would be used only if there was a substantial loss of whole blood. Packed cells are given much more frequently, particularly to treat anemias. If we just need to replace volume, we can use blood expanders. This will replace the fluid. Things like saline, albumin, or plasma work. Then there are other blood components that can be transfused, things like platelets, factor VIII, even white blood cells. If we're going to transfuse blood, we need to know a little bit about the human blood groups. These are genetically determined. They're based on antigens or proteins that are found on the surface of the red blood cells. There are over 90 known blood groups, but the ones that give us problems with transfusions are the ABO and the RH blood group. In the ABO system, there are two antigens, the A antigen and the B antigen. If you have one of the antigens, the body will naturally produce antibodies to the missing antigen. So that if someone is group A, that means they have the A antigens on the surface of their red blood cells, and they will naturally produce B antibodies. Someone who is group B will have B antigens on the surface of their red cells. They will naturally produce A antibodies. Someone who is group AB has both A and B antigens on the surface of their red blood cells and therefore will produce no antibodies. And someone who is group O has no antigens, that O is really a 0, and they will produce both anti-A and anti-B. The interaction between the red cells and the antibodies cause clumping of the blood cells, or agglutination. So here you see someone who has A antigens because they reacted with the anti-A serum, and they have B antigens because they reacted with the anti-B serum. This is someone with type AB blood. Here, the cells only reacted with the B antiserum, so this is type B blood. The cells only reacted with the A antibody here, so this is type A blood. And since there's no reaction with either antisera, we have type O blood. The Rh blood group has 5 major antigens: big C, big D, big E, little c, and little e. The most antigenic is D, and when we say someone is Rh positive or Rh negative, it's because of the D antigen. About 85% of the population in the United States is Rh positive, meaning the other 15% are Rh negative. Rh positive people have the D antigen on their red cell surface. Rh negative people do not have the D antigen on their red cell surface. We do not get naturally occurring antibodies to D, so if you are D negative or Rh negative, you do not have anti-D antibodies. The only way you'll produce antibodies to D is if you are exposed to Rh positive blood. Hemolytic disease of the newborn, or HDN, also is known as erythroblastosis fetalis. This can occur if an Rh negative mother gives birth to an Rh positive baby. Now, normally there is no mixing of the mother's blood with the baby's blood during the pregnancy, but at the time of delivery, some of the baby's blood may get into the mother's bloodstream. Because the baby is Rh positive and the mother is Rh negative, the mother will produce antibodies to the Rh positive blood, these anti-D antibodies. These are small antibodies that will cross the placenta during subsequent pregnancies. The D antibodies cause hemolysis. The next baby, if it's Rh positive, will have these antibodies passed to it from the mother. The baby's blood will be hemolyzed. The baby is born with an incredibly high bilirubin, and bilirubin can damage the nervous system, so we need to get rid of that as quickly as possible. And they may need a transfusion themselves when they're born because they have too little blood to carry oxygen. This is prevented today by giving a substance called Rogam. They used to give it within 72 hours of birth, but now they tend to give it during the pregnancy. If we give someone blood or blood products, we are said to have given them a transfusion. Theoretically, the universal donor is O negative because someone who is O negative has no A, no B, or no D antigens on the surface of their red cells. They wouldn't react with anybody's blood. The universal recipient theoretically is someone who is AB positive. The AB positive person has no antibodies. They have all of the antigens, so they have no anti-A, no anti-B, no anti-D. Therefore, they can take any kind of blood. In actual practice, you need to give group and type specific blood, if at all possible. ABO incompatibility can cause agglutination of the donor cells. These little tiny clots can act to block blood vessels. Rh incompatibility can cause hemolysis of the donated cells. In this case, the extra bilirubin may put stress on the kidney and cause kidney failure. Either of these is considered a transfusion reaction. Whenever a transfusion reaction is suspected, the transfusion is immediately stopped. Sometimes we just need to restore blood volume. A low blood volume leads to a low blood pressure, and over time, if it gets low enough, you go into shock and die. We typically replace with fluid, not blood, things like isotonic saline or Ringer's lactate. These will replace the volume, but they do not replace the oxygen carrying capacity of the blood. There has been some work done with artificial blood substitutes. These would not have the problem of being antigenic and would have the ability to carry oxygen. The blood can be a great way to check out what's going on with several systems in the body. A white blood cell count with a differential can give you the number and type of white blood cells, so you can determine whether it's a bacterial infection, a viral infection, or an infection at all. Coagulation studies can be done to see if the coagulation factors are present in the appropriate quantities. A CBC, or a complete blood count, will do a count of your red cells, your white cells, your hemoglobin. And a comprehensive metabolic panel will look at all of those metabolites in your blood, glucose and the various liver enzymes and muscle enzymes, to see if you're overall healthy. As we age, we can expect to see some changes in the blood. First of all, as soon as we're born, we begin to replace our hemoglobin. As a fetus, we have hemoglobin F in our blood. Hemoglobin F has a greater affinity for oxygen than hemoglobin A. Since as a fetus we're getting oxygen transfused from the mother across the placenta, we need to make sure we capture all the oxygen that comes across. Once you're born and you start breathing air, you can convert over to hemoglobin A. Because hemoglobin has to be processed by the liver, sometimes newborns have a little excess bilirubin left over, making them appear jaundiced. As we age, we're more likely to develop chronic leukemia, either as a result of insults to our DNA or as a result of having something called the Philadelphia chromosome. Thromboembolic diseases are a little more common as we age. Again, the longer we live, the more likely we are to have done some micro tears in our blood vessel. Those micro tears will be scar tissue, which will be a little rougher than our standard endothelial lining. Platelets like to cover up rough spots. Anemias may develop either because of gastric problems, the inability to produce intrinsic factor and therefore use B12, or dietary problems where you may not take in enough iron or B12 in your diet. Most problems with the circulatory system are the result of heart, blood vessel, or immune system problems, not problems with the blood.